Okay, looks like we have a good number of attendees here in the room and also online. So I will go ahead and start. Welcome you to the Genomics and Health Disparities lecture series. My name is Leonardo Mariño. I am a Statman investigator at the National Institutes of Minority Health and Health Disparities here at the NIH. I would like to welcome you to the 32nd lecture on the NIH Genomics and Health Disparities lecture series, which began in May of 2015. This series aims to highlight opportunities of genomics research to address health disparities. This series is sponsored by the National Human Genome Research Institute, the National Heart, Lung, Blood Institute, the National Institute on Minority Health and Health Disparities, the National Institute of Diabetes and Digestive and Kidney Diseases at the National Institutes of Health, as well as the Office of Minority Health and Health Equity at the Food and Drug Administration. The speakers have been chosen by the series co-sponsors to present the research on the ability of genomics to improve health for all populations, and often the challenges in making sure these improvements are accessible and applicable for all populations. The speakers in the series approach this problem from different areas of research, including basic science, population genomics, translational and clinical and social science research. I'm pleased to welcome Dr. King Jordan. He's joining us from Georgia Tech, and I will briefly introduce him. Dr. King Jordan is a professor in the School of Biological Sciences and also the director of the Bioinformatics Graduate Program at the Georgia Institute of Technology. So I've met King back in, this was, I think, 2002 or 2003 back when we were doing research at the NCBI. I remember that at some point King decided to go to Colombia and learn Spanish. He's now fluent in Spanish. He has embraced the Latino culture. and so it has been a pleasure for me to work with him over the years. Now, after the talk, I will facilitate a discussion with virtual attendees and attendees here. So please submit your questions at any time via the Q&A box. And please welcome Dr. Jordan.
Okay, Leo, thanks a lot, and appreciate it. Thank you to the organizers. Thanks to all of you for coming. Thanks to those online. Okay, so today I'm going to talk about our research on genetic ancestry, population structure, and health outcomes using the All of Us research program cohort. First and foremost, a disclaimer, I am not a member of the consortium, as you just heard from Leo. I'm from Georgia Tech in Atlanta, but I'm very delighted to be collaborating with people from All of Us to do this important work. So this is what I'm going to talk about today. I'm going to start by talking about diversity in the All of Us research program. Then I'm going to do some basic definitions. I know we have a diverse audience here with respect to scientific background, so I'm going to talk about what is genetic ancestry and population structure, which are I'll try not to be too pedantic. I am a professor, but these are the key themes of the talk, and I'll touch on those. And then I'll talk about kind of a hybrid of our algorithm development work to develop algorithms to infer genetic ancestry at biobank scale on huge data sets of hundreds of thousands of participants. And I'll talk about how we are applying those algorithms to the All of Us cohort. And then finally, I'm going to talk about the relationship between genetic ancestry and health outcomes. And this is related to the theme of this symposium of health disparities. And I'll talk about two different techniques. One is admixture regression, and the other one is admixture mapping. So I think by now, most of you are probably familiar with this problem, the genomics research gap. So it happens that the vast majority of study participants in genetics and genomic studies have European ancestry, vastly disproportional to their population on the global scale. And this has the potential to exacerbate health disparities by limiting the reach of genomic medicine. And results from that are derived from studies on European ancestry cohorts are less accurate for non-European ancestry individuals. So this brings us to all of us, and they have boldly stepped into the breach in an effort to try to close the genomics research gap. Again, I am not a member of the consortium, but they have done a really, really amazing job. And what they did, what they set out to do, and they succeeded wildly, at least in my estimation, is that they wanted to enroll a diverse cohort of participants from communities that have been historically underrepresented in biomedical research, including but not limited to racial and ethnic minorities, in support of more equitable genomic medicine. And so what you see over here on this slide is just a comparison of the percentage of participants in all of those shown in gold compared to the recent US Census. And so unlike almost all other studies prior to all of us, they have been able to get at least as much, and in some cases, slight overrepresentation of populations that were historically left out of genetics and genomics research. Now, when we started this work together with all of us, this was the result of our very preliminary analysis, which was published as a part of the flagship paper that came out in Nature. And some of you may know, it depends maybe how much you're on Twitter, but this generated a lot of controversy. And the controversy, there's really sort of two aspects to the controversy. So one aspect was a methodological issue, which was, what is the best way to represent really highly multidimensional genomic data in a reduced dimension set, in 2 dimensions in the case of UMAP? And there are algorithm developers that are very, very passionate about these issues. So there was a lot of discussion about that. But the reality is that's kind of a niche computer science issue. And for ourselves, we are very much agnostic with respect to methods. So we feel like we don't really have a dog in this race in terms of which methods are best for dimension reduction. But the more important I think the part of the controversy that really resonated was the comparison of participant self-identified race and ethnicity, which are socially defined and distinct from genetically inferred ancestry, and I'll talk about that more, with genetically inferred ancestry. So when you combine this with the UMAP, and the critique there is that unlike PCA, and I'll show you lots of PCA today, principal component analysis, UMAP, the aim of UMAP is to define and visualize clusters. So by visualizing clusters or emphasizing clusters in the data, and then overlaying how the participants and all of us self-identified their race and ethnicity, this gave the impression that there were very discrete groups, that there was more discrete ancestry than there is in reality. Now, having said that, what we do in our lab, and this is what I'll talk about today, is we don't deal so much with ancestry group, but rather we deal more with continuous ancestry. So we infer and model genetic ancestry as a continuous variable with respect to fractions of ancestry for individuals. And this gives us a lot more resolution than you can get when you try to maybe shoehorn participants into groups which are very broad and heterogeneous. Because when you have this continuous ancestry, you can really see that while there are clearly group-specific patterns of ancestry, everyone and all of us is admixed to some extent, and there's lots and lots of variation within groups. Okay, so I'm not going to belabor that. That's not what I'm here to talk about. But what this allows us to do is kind of segue into what we were trying to do in the first place, which is a much more detailed and in-depth analysis of the cohort. So we just heard from Leo, and I just want to say from the outset that all of this work is done in collaboration with Leo's lab at NIMHD. I mentioned that I'm not a member of the consortium, but Rob Miller at the Morehouse School of Medicine is, and he's the one that introduced us to the consortium. And the main protagonist of this story is Shivam. Shivam's a PhD student in my lab, and he's the one that has done the bulk of this work. So he's the one that's really, really behind the work. Okay, so what we wanted to ask with respect to the ancestry is, given the really wild success of all of us in recruiting individuals and enrolling individuals that have been left out of research, what are the implications of the racial and ethnic diversity of the cohort for the genomic diversity, specifically genetic ancestry and population structure? In other words, to what extent has all of us, in fact, closed the genomics research gap? Okay, professor time, a little bit of definitions, just very briefly. Population structure, taken from Wikipedia, don't judge, but it's actually not a bad definition. So this is simply differences in allele frequencies among different groups or populations within the same species. So in randomly mating populations or panmictic populations, you don't get any differences in allele frequencies across groups, and really no groups that you can resolve. But human mating, like most other mammals or most other sexually reproducing species, is almost never random. So population structure occurs in humans and other sexually reproducing species due to non-random mating, and non-random mating occurs based on geographical distance, physical barriers, and even cultural differences in modern cosmopolitan populations. And so population structure, in turn, links us to the concept of ancestry. It's closely related to ancestry. And with continuous ancestry, again, we can measure how much of an individual's genome comes from a set of defined groups or populations. This is supervised genetic ancestry inference. I'll talk a little bit more about what I mean by that. And we do that by studying genomic variant data, looking at population structure, and then leveraging that population structure to infer genetic ancestry. And all of this is done by comparison between reference individuals. So these are samples from global reference populations, which are in turn compared to query individuals. And query individuals, in the case of this story, is the All of Us research program participants. Okay. Ancestry, in the broadest sense, just refers to the geographic origins of an individual's ancestors, and it impacts health and health disparity in a number of ways. And you can think about ancestry as socially defined with respect to race and ethnicity, or genetically inferred with respect to ancestry. And so genetically inferred ancestry, which is what we look at in the lab, is a characteristic of the genome. It's written in your DNA. So unlike race and ethnicity, is not a socially defined variable or parameter. Because of that, because DNA is a digital code, we can infer genetic ancestry objectively and precision. We can infer it to a large extent independent of the social dimensions of race and ethnicity. We can infer it as a categorical or continuous variable, and I'm really going to emphasize over and over again, we're looking at genetic ancestry as a continuous variable to get increased resolution. You can look at different levels of resolution with respect to the genome, so you can infer someone's ancestry over the whole genome, but you can also look at location-specific patterns of ancestry. In other words, you can infer ancestral origins for each haplotype along the genome. And then finally, you can look at different levels of relatedness in terms of continental or more fine-scale subcontinental ancestry. Now, this finally brings us to the analysis of the data. So this is an analysis of controlled tier dataset version 7 of All of Us. I hear on good authority that version 8 is coming very, very soon. We're looking for that, looking forward to that. But version 7 has, as you can see, lots and lots of data, right? And so this is both a blessing and a curse. I mean, it's amazing to work with data sets of this size, but it presents a lot of challenges. So we, and the we is Shivam, and Shashwat before him, start out with just over 400,000 individuals, then takes individuals with unrelated genomic data. So I should mention that these are the genome-wide genotype data, but if you use the whole genome sequence data, you get essentially the same results. And then he slices and dices these a number of different ways. We're going to talk about the relationship between ancestry and geography with zip code data, unsupervised, supervised ancestry inference, and then ancestry inference at different levels of relatedness. All right, so the first thing is unsupervised population structure, and here I talked about dimension reduction, the use of UMAP, the controversy that came. But before you even get to UMAP, UMAP works on PCA, and PCA, principal component analysis, is a much more standard and widely used method for dimension reduction in genomic data. So what we're going to do is we're going to do genetic PCA on the data and then perform unsupervised clustering to assess the extent of population structure in the All of Us cohort. So here is the first view that you get. And I'm showing you the first two principal components. We typically go out to about 20, but in a very diverse data set of this kind, you hit diminishing returns very quickly. In other words, most of the variation in the data set is explained with the first few components, and the other ones don't explain as much. So you can get a pretty decent view looking at the first two, and I'll show you a little bit of the third one. But this looks, at first glance, this PCA projection, like a continuum of diversity in the All of Us cohort, okay? And this is very much consistent with this paper that came out in Science. This was a perspective piece that dealt, before the controversy about this same issue. There's a controversy about genetic ancestry. How do we get it right? The stakes are high. This is very important. And they showed this principal component analysis, and this is I don't know if you say BioME or BioMe, but it's a big biobank in New York, I think at the Icahn School of Medicine. A lot of you probably know about it. Not as big as All of Us, but it's also very, very diverse. And the point of this was to show the relationship between the individuals from the biobank in gray, just as I've shown for All of Us, and the global reference populations that are used to compare to biobank participants to infer ancestry. And the point they made is that these clearly delineated continental ancestry categories, what they call islands of colors, are a byproduct of the sampling strategy, right? You go to Africa, you go to Europe, you're sampling from different parts of the world, and you get the appearance of these discrete clusters, whereas if you look at the individuals in the biobank, these are sampled from the New York area largely, you get what they call this continuous sea of gray. In other words, the appearance of population structure is actually an artifact or an illusion of sampling, okay? Now, I'm saying not so fast, because so all of these methods, someone once said, there's no solutions, only trade-offs, right? And that's definitely true for genomic data analysis, right? When you talk about data dimension reductionality, there's no one-size-fits-all perfect method. And so while UMAP has its disadvantages, PCA has its disadvantages as well. And one of the disadvantages of PCA, and this is really most relevant when you have very large, complex cohorts, is you have lots and lots of people and the points are over-plotted. In UMAP, you have the opposite problem, because every individual gets a point in UMAP space, and that can artifactually blow up clusters. So what Shivam did is he said, okay, how many neighbors does every individual in PCA space have? And then you can see the implications of these over-plotted points in PCA space, because in fact, it's not just this continuous sea of gray, but rather there are very clear discrete groups of genetically related individuals in the cohort. This is just PC1 against PC3. You can see that in a different way. And then another cool thing he did is he did kernel density estimation, and this essentially is just another way of looking at the same thing. And again, what you see is, rather than a continuous sea of gray where there is no apparent population structure, there is in fact a lot of population structure that can be perceived if you can somehow drill down and look at the density of the plots in PCA space. And if you try to cluster these, and here we talked about lots of different methods, you can use UMAP, you can use, this is HBD scan, you can use k-means, you can use KNN. It doesn't matter what method you use to cluster, you get very coherent and distinct clusters. But of course, in between those clusters, you see lots of individuals that don't group to any specific cluster, okay? And then there's a statistic called the Hopkins statistic. It ranges from 0 to 1, 0 being totally random, 1 being very, very clustered, and this gives a value of about 1. So the data are highly clustered, and there are natural clusters of genetic similar individuals. So in other words, there's lots of population structure in all of us. It's not just an artifact of sampling, but rather it reflects the diversity in the population, and that in turn allows us to do genetic ancestry inference. So without getting into the gory details of the algorithms, I just want to mention that there's great methods out there for genetic ancestry inference: admixture, RFmix, chroma painter, but all of them are over 10 years old, and all of them were designed to work on 10 or 100 or at most a couple thousand samples. They work great, but they simply do not scale well to biobank-sized data sets of tens of thousands or hundreds of thousands of samples. Given that challenge with Leo's group, we developed this method called RAI, which works on principal component analysis. It takes global reference populations, does PCA on those, along with the query individuals. In this case, these are individuals from Colombia, but this could be all of us participants. And then it uses an iterative process to estimate average eigenvectors or average principal component vectors for the reference populations. That is the slowest part of the algorithm, although it's not slow compared to older algorithms. And then it really gets its speed from non-negative least squares. It does a little bit of linear algebra to convert the reference population ancestries into estimates of the percent ancestry for each reference population component. In other words, admixed individuals from the all of us cohort are modeled as linear combinations of ancestry components from reference populations. That's how RAI works. It gives you basically the same results as admixture, RFmix, whatever your favorite older method is. It's very, very accurate, but the big thing is that it's fast, right? So it runs in about 44 minutes after you do PCA, compared to 37 hours on admixture, and this is on UK Biobank, about 500,000 individuals. So this allowed us to infer ancestry for the All of Us program, and the way that we're doing, now we're moving from unsupervised, which was the population structure, to supervised, which is the ancestry inference. We compare All of Us participants to global reference populations from the Thousands Genomes Project, from the Human Genome Diversity Project. Again, here I'm emphasizing, or perhaps belaboring, that we are inferring ancestry as a continuous variable. One of the things that All of Us does, and this is very useful for stratifying cohorts for association studies, is they provide categorical ancestry groups, or genetic similarity groups, they refer to them as. And these, so these are the All of Us groups. They are African, admixed American, East Asian, European, Middle Eastern, or South Asian. But these groups are very, very similar to how individuals self-identify their race and ethnicity. So the African ancestry group, which is a supervised ancestry inference on genetic PCA data, again, these are the data from All of Us, not from our group, irrespective of how people self-identify, nevertheless is about 91% of the individuals in this ancestry group self-identify as Black or African American, 88% in the admixed American self-identify as Hispanic or Latino, and so on, okay? So you still have these broad, rather heterogeneous groups, and the continuous ancestry provides additional resolution. So this is what that looks like. If you kind of think back to the BioME example I showed, this is very similar. So you have these coherent reference populations, and then you have these very dispersed or relatively dispersed All of Us participants, bearing in mind that the gray kind of doesn't show you the coherent clusters, and from that we can model individuals as linear combinations of those ancestry components. So here we have the 7 clusters, which are generated by unsupervised clustering of the PCA data, and then here we have their ancestry components. And if you are not used to this kind of plot, this is like a stacked bar chart. You see the percent ancestry on the y-axis. There's lots of participants, so it's hard to see each one, but each line is a participant, and it shows the combination of ancestry components for all of the groups. So right away, you see very distinct ancestry patterns between the clusters of individuals in PCA space, not surprising given how the method works, but lots and lots of variation within groups. And just by way of comparison with the All of Us genetic similarity, or the All of Us groups, these correspond well for the most part, right? So the AFR group, or the African-like group from All of Us, corresponds to cluster 1. Again, this is supervised. Ours is unsupervised. All the others correspond well. Where it doesn't is admixed American, which, if you know anything about the population, the history of these populations is not surprising, right? So you have cluster 2, which goes along the axis goes along the first PC, and these are individuals that have European, African, a large African ancestry component, and indigenous American or Native American ancestry here. And then you have AMR2, which goes along the PC2 axis. And these individuals have the same ancestry components, albeit in different proportions, more European ancestry, a very high secondary component of Native American ancestry with African ancestry there. And you can look at how this varies across geography. So this is when you look at all of the data, and you can begin to see patterns, differences across the country. More African ancestry in the southeast. You can see more American ancestry in the west, more Asian in Hawaii. If you take out that European ancestry component, then the geographic patterns become a little bit more apparent. You can see a lot of admixture in Puerto Rico and Florida and Hawaii and Texas, for example, but the main patterns are kind of the same. And then finally, the last thing on ancestry, and this was really quite striking to us, is that we stratified the all of us cohort by participant age, and then we calculated genetic admixture entropy. This is just Shannon's entropy, but it is calculated on the ancestry components. So the higher the value on the y-axis, the more admixed or the more diverse, genetically diverse the individuals are. And the younger the participants are, the more admixed they are, they're more ancestrally diverse, and there's a very, strong, almost monotonic pattern there. Okay, so now moving on to health. So the question then becomes, what do these patterns of ancestry and admixture mean for health outcomes in the all of us cohort? And the two techniques we use, again, are admixture regression and admixture mapping. So for admixture regression, we use a phenome-wide association study of continuous ancestry fractions with health outcomes, okay? So all of us provides participant ICD-9-CM and ICD-10-CM codes. So these are disease and health codes gleaned from electronic health records. We map them to phi codes. Phi codes have a lot of advantages for deriving case control cohorts from electronic health records. And for the uninitiated, if you're not familiar with this term phenome-wide association study, what it means is you do association with all of the outcomes that you can get your hand on. So in this case, it's all of the health outcomes. And so there's close to 2,000, but for those that have greater than 100 cases, which gives us statistical resolution, we're able to get 1,674 phi codes. And then what we're doing is we're modeling, so this is logistic regression, cases and controls, ones and zeros, each phi code by the fraction of ancestry, and we're focused on the 3 largest ancestry components in the cohort, which are African, American, and European. And in all of our models, we control for age and sex. So if you look at African ancestry, so this is a volcano plot. So you see the effect size on the x-axis. You see the level of statistical significance on the y-axis, negative log transform p. So the higher you are, the more significant you are. And what you can see is a number of both positive and negative associations with African ancestry. And I'm just highlighting some of the largest ones here. There's HIV, there's substance addiction disorders, alcoholism, hypertension, and then on the other side, there's a number of skin conditions. There's also type 2 diabetes and diabetes here. Sometimes these phi codes are nested, so you'll have a higher level 1 that includes the lower level 1. If you look at American ancestry fractions, you also see a number of associations. Diabetes in particular gets quite high. Sepsis, other glucose. But again, you see this kind of mirror image where you see both positive and negative associations with ancestry. And then finally, with European, if you can recall back to the African, it sort of flips, where you have a lot of positive associations with skin conditions, skin cancer, anxiety disorder has the highest p-value. There's also a number of cancers here, and then some of the ones that were positive in the other ancestries flip and are now negatively associated with European ancestry. So to summarize all that, another nice thing about phe codes is you have categories. So they have 34 categories of related diseases. So you can look at the enrichment. This is like a gene set enrichment analysis, but with phe code categories for the different diseases, seeing both the negative and the positive associations. And it's quite striking that over 1,200 of these 1,674 diseases, so 73%, almost 3/4, show significant ancestry associations at a Bonferroni corrected p-value. However, very, very important caveat is that these admixture regression associations may or may not represent direct genetic effects, right? There's lots of other environmental and non-genetic factors that contribute to health. If those environmental factors also vary along the axis of ancestry, what you might be seeing here is indirect effects whereby the ancestry is essentially tagging some environmental effect, okay? So the fact that you have these ancestry disparities really, in essence, just highlights, or the ancestry associations rather, just highlights disparities, but it is not a statement of causal effect. Okay, that's very, very important to keep in mind. And then we're just starting to get into this, so the future work for us is now to take the genetic ancestry and analyze it side by side with environmental exposures, and we are particularly interested in social determinants of health. So Sonali Gupta, working in the lab, recently developed a composite socioeconomic deprivation index from the All of Us survey data. So this takes information about income, education, employment, health insurance, and housing, and gives you a quantitative value. The higher you are, the more deprived you are. And this is very preliminary, so I just want to highlight that when you include socioeconomic deprivation in these models that include ancestry, the effects of ancestry become attenuated, right? So you see a big attenuation for HIV, a very, very big one for substance addiction disorders, and other ones. So there's almost always an attenuation, right? So this is consistent with that idea that these are not necessarily direct genetic effects, but rather they are tagging something else. And so the future for us, the near future right now, is trying our best to find out what that other thing is. And the great thing about All of Us is that they have lots and lots of data that you can use to do that. And so the job of the students in the lab, and Vincent back here is working on that too, is to pull out the survey data from All of Us and then develop these quantitative metrics, these indices that we can plug into our models. And then finally, very last thing is admixture mapping. And so the hope here is that by narrowing the focus from genome-wide patterns of ancestry to locus-specific ancestry patterns, we can get closer to, I want to be cautious about this, these are observational data, but we can get closer to causal inference, okay? Why is that? That is because what admixture mapping does, and this is interesting because this is actually an old technique, it's a pre-GWAS technique, but it's kind of making a comeback with respect to genomics and health disparities. But anyway, with admixture mapping, you associate the local ancestry dosage, in other words, the haplotype-specific ancestry, with the health outcome, right? So here you have a scheme, so these would be two-way admixed individuals, doesn't matter what the ancestry components are, dark green and light green, and when you compare cases for whatever your disease of interest is to controls, and you actually don't need controls because the genomic background can do it for you, an ancestry association is just a peak whereby there's a region in the genome that has an anomalously high or low ancestry contribution in the cases, okay? So if any of you are familiar with Mendelian randomization, this is essentially the same thing. It's Mendelian randomization, but using, instead of SNP dosage, local ancestry dosage, right? So with Mendelian randomization, the random part happens due to the random segregation of chromosomes during meiosis, and so there is a decoupling of that relationship between ancestry and the environment at meiosis, and that's really the power of admixture mapping. So we hope that this is more likely to capture direct effects of ancestry on health outcomes. So here we have a smaller cohort, two-way admixed, African and European ancestry, close to 40,000. Same exercise in the sense that we're going phenome-wide, but now instead of mapping or modeling rather genome-wide ancestry, we are modeling the ancestry dosage, where the ancestry dosage is 0, 1, or 2 copies of either the African or the European haplotype, right? So this is just like GWAS, but the dosage is not SNP allele dosage for biallelic variants, but rather it's ancestry dosage. There's a lot of talk about how to control for multiple testing in admixture mapping studies, because those blocks are long, and you're actually, when you do an admixture mapping study, you are performing fewer independent statistical tests than you are when you do a GWAS. So the field has kind of converged on a genome-wide admixture mapping Bonferroni correction of 1 to the E minus 5. So, you know, if you want to find results, that's kind of nice because it's a little bit more of a permissive, but we have to take into account that we're analyzing 1,000 phi codes. So when you add that in, you get back to the old faithful 1E to the minus 8. And so these are very, very preliminary results coming out of Leo's lab. And so what you're seeing here, I'm just going to show you a number of these plots. So again, these look like they are Manhattan plots. They look like GWAS Manhattan plots, but they're not GWAS Manhattan plots. They are admixture mapping Manhattan plots, or Miami plots rather, because you have the mirror if you prefer Chicago. We like Miami better. We're from the South. But here you have the African enrichment, and then here you have the European enrichment. So this first result is for sickle cell. Now this is not at all surprising. So we just take this as a positive control. It's just a proof of concept. It's also interesting because we're looking at ancestral haplotypes, but they appear to be able to tag rare, in quotes, sickle cell variant is not that rare, but you know, somewhat rare coding variants, and you'll see in coming slides. So this is kind of like a proof of concept. And then we get to hyperlipidemia, and this is really interesting too, because this PCSK9 gene, knockouts of this gene, natural knockouts were found, I don't know, more than 10 years ago to give people very, very low, insanely low cholesterol levels. And then there's been a mad dash on since then to do drug development on that gene. That was found in an African-American woman. It was written up in the New York Times. So it's interesting that the European ancestry, not the African ancestry, is enriched for hyperlipidemia, suggesting the possibility that these natural knockouts are found on an African ancestry background, consistent with that old story. And then there's a number of interesting results on diseases of white blood cells. So again, here we're looking at an enrichment of European ancestry, which maps to the Duffy antigen gene, ECKR1. This is the gene that evolved for malaria resistance, and it is associated with elevated white blood cell count. But then if you flip that and you get to decreased white blood cell count, then the ancestry association flips to African ancestry, and it's known that the mutation in this gene, which confers resistance to malaria, is associated with low white blood cell count, but it's benign neutropenia in African Americans. So the admixture mapping picks that up too. So like I said, this is very preliminary. This is where we are. So we have fewer cases consistent with the idea that going genome-wide gives you a lot of associations, but essentially what you're finding there are disparities which may or may not be attributed to direct genetic effects. With the admixture mapping, we had the randomization at meiosis, which hopefully gets us closer to causal inferences. So we have 6 phi codes with very strong associations, p minus 1 to the minus 8, less than 1 to the minus 8. We have 7 with marginal associations. So we're looking forward to the release of version 8. We're hoping we can power up these studies with the additional data that's going to come out on version 8. So we see ancestry-specific effects on hyperlipidemia and white blood cell count. We want to include more diverse ancestries in these cohorts, so this it's relatively easy to do this two-way because it's just like a GWAS with biallelic variants. It's a little bit trickier. We're trying to work out multiple ancestries, which would be like doing a multi-allele GWAS, it's doable, it's just not quite as straightforward. But what we really want to do is get into some fine mapping and conditional analyses. So these admixture blocks are rather large. That's why I mentioned at the beginning, it's an old technique, so something that people were using before. We want to now go into those large blocks, do fine mapping, and when we find, now it would be SNP allele dosage against phenotype, when we find those associations, see if we can do conditional models whereby we regress out the effect of ancestry using variants that show ancestry-specific allele frequencies in different populations. In other words, can we attribute the observed ancestry effects to one or a few SNPs? So that's what's coming on the horizon there. So in conclusion, so all of us has really done an amazing job of recruiting underrepresented racial and ethnic groups, and as such, it has yielded a very genetically diverse cohort. There's lots of population structure. This is much more than this idea of a mere continuum of diversity. So there's kind of two things going on at the same time, right? We can model genetic ancestry as a continuous variable, thereby avoiding the issues of grouping together people in very heterogeneous groups and get more resolution in our models. But at the same time, there are discontinuities in those continuous distributions. So both things are kind of going on at the same time. Genetic ancestry in the All of Us program changes over geographic space and time. And one big message I want to get across today is that genetic ancestry matters for health. There's more than 1,000 genome-wide ancestry health associations. This includes direct and indirect effects of ancestry. And then there's a few very preliminary, interesting local ancestry health associations that are much more likely to implicate direct genetic effects. And finally, I just want to close here with making the case for the relevance of ancestry for these kinds of studies. I know in the health disparities community, there's lots of debate about the role of genetics, and there's concerns there. I get that. But the message I'm delivering is that these genetic differences between population groups are real. They exist, they matter for health outcomes, they matter for health disparities. They're important for both research in genomics, but also genomic medicine, especially as it relates to the reach of genomic medicine. So pretending that these differences don't exist or deliberately obscuring them will ensure that the groups who stand to benefit the most from precision medicine, in other words, the groups that already have the most health disparities, will gain the least. This will exacerbate rather than ameliorate health disparities. And this is the case that Leo and Eliseo and I made in this little letter to Nature here. And this issue has been brewing for a long time, right? There's nothing new under the sun. So this is a book by Theodosius Dobzhansky. It's from 1973, right? Before we could even sequence DNA. It's genetic diversity and human equality. And what Dobzhansky was trying to do was make a very clear case about the need to decouple, to disentangle, to separate conceptually the idea of the empirical observation of diversity. Diversity is out there, genetic diversity is out there, from the moral and ethical notions of human equality, human dignity, equal rights, and really, disentangle those. And too often, those things become conflated, and that's a big part of the controversy, right? So you have the mixing of those ideas, and those really need to be separated out because, as Dobzhansky says, diversity is an observable fact of nature, while equality is an ethical commandment, okay? So, thank you for your attention. I'll stop there and take questions.
Okay, so let's go ahead and start with questions from the audience.
I have a,
Yeah, I think the back is the one that works, yeah.
Hi, I'm Jackie. I'm a postdoctoral fellow here in Neil Hanchard's lab. I have a comment and then kind of two related questions. So when I was doing my graduate work, we did essentially the exact same study using Vanderbilt's BioView and found a lot of the similar results. But one of the things that we did not do was include the social determinants of health.
Right.
And that upset a number of people when we presented the results, because as you said, there are things that pop up that are just along for the ride because they are involved in socioeconomic
That's a good way of
status.
saying it, by the way. Yeah.
Yes. Like the pregnancy complications, for example, that was something someone took a lot of offense with, because you would see that African ancestry is enriched for pregnancy complications, whereas European ancestry is de-enriched for pregnancy complications. That's more likely to do with socioeconomic status than it is about genetics. So that's very good to see, and I'm glad you're not in the same trap we were, because that helps to really tease apart when I tell people genetics aren't everything, socioeconomic status matters more in a lot of cases. And then, so that was my comment. My next question was, have you guys started on modeling like ancestry interactions, like portion of, American ancestry and European ancestry, and how those interact with each other for disease?
That's a very interesting question. I'm going to start with your second question and then go back. I want to just basically double down on your comment, so thank you for that. We haven't looked at ancestry interactions, ancestry to ancestry, because they are dependent, right? So if you have a total ancestry fraction of 1, then the individual components are dependent on each other, so that's tricky statistically. But what we have looked at, if you look over your left shoulder, you'll see Vincent sitting in the back there, who has looked at ancestry by socioeconomic deprivation interactions for type 2 diabetes and gotten some interesting results. You published that, what, last year? And so I'm talking really all about ancestry today, but one of the great things about all of us, and now this is kind of bridging to your first comment, is all of the non-genetic data, the combination of the genetic and the non-genetic data, much of which, the kind that interests us anyway, are found in all of these surveys. So what the heavy lifting, the students' heavy lifting, is to kind of take those survey data and then figure out a way to quantify those data, because you have lots and lots of survey questions and there's lots of multi-collinearity there. So that's what we're really, really pushing on. So I totally agree with you. And Eric Green made this point on Twitter earlier this year. The NASEM report on population descriptors made a very strong statement about the need to include non-genetic factors and to collaborate with people that are working on social determinants of health, all of which I agree with totally. And that's what we're trying to do. Again, I just barely scratched the surface here. We've done a lot of that. But I would also like to make the case that the converse is true as well, right? So the flip side of the coin is that if you are interested in social determinants of health, especially as they relate to health disparities, if you model social determinants of health without including genetic principal components, like in GWAS, it's very standard to include genetic principal components to control for population structure, you are very likely to get confounded results. So confounding is a two-way street, right? If you only do genetics, it's definitely going to be confounded by non-genetic factors. If you only do non-genetic factors, and we found this, Vincent, again, this is Vincent's work, has shown that the effect size estimates of social determinants of health really change when you include genetic principal components. And this is, very standard in the GWAS field, but not at all standard in the social determinants of health field. And if you do a PubMed search and you look at social determinants of health and health disparities, and then health disparities in genetics, you get big numbers on both, but the intersection of that Venn diagram is really, really small. We want to grow the intersection of that Venn diagram. We agree that that's critically important. Yeah. Good, thank you.
In the spirit of two questions. First, I do appreciate putting the density function into a principal component plot that has 300,000 people. I'm not sure that means it's not a still sampling problem. And imagine you've got one of these gray areas that is not in one of your pictures of density, and just imagine you had 10,000 identical twins that you sampled. I know you excluded related. That would show up as a density plot based on sampling more than anything else. So I'm not sure I buy that you've totally decoupled it with sampling, but it allows you to view something in the samples that you can't see otherwise. Am I thinking about it the wrong way as the function?
No, I mean, and it's interesting when you think about sampling, right? So one of the things I didn't say is that all of us, like almost all large biomedical databases or population biobanks, is a non-random sample of the US population. If you're talking about something like NHANES, where they really drill down and they have this statistical sampling procedure, then the results of any analysis on NHANES you can extrapolate out there externally valid to the US population. That's not the case for all of us. Having said that, because of the emphasis on partnering with HBCUs and Hispanic serving institutions, they've done such a good job. If you think back to that first plot on comparing the census to the US population at enrolling individuals from groups that have been left out of research, that the demographics of all of us actually reflect pretty well the demographics of the United States, with the important caveat that it's a non-random sample. So, is there a sampling issue? Yes, but it is a historical sampling issue, right? So the US population is an historical sample of immigration, forced immigration, the transatlantic slave trade, the people that were here before those who immigrated came, and so on, right? So if you look at the US or the UK or any modern Colombia, any modern cosmopolitan population, you have a non-random sample of global genomic diversity based on the pattern of the history of the country. So what the PCA, the fact that the density doesn't reflect a sampling bias per se, it does reflect the unique demographics and history of the country, but it captures that pretty well. Yeah.
Yeah, I think we were saying the same thing then.
Yeah, okay.
And then the other question was to go back to drill into the PCSK9. Have you had a chance to look in GTEx to see whether there are associations with expression, and can you subtract out all the nulls?
These data are 2 days old. I'm very excited to present them. The answer is no, and that's a great idea. We really want to drill down. First, we want to reproduce on version 8, and we want to drill down into those blocks and see what's going on. And the nice thing, on the one hand, you get 1,000 associations with genome-wide ancestry, and that excites us because we're into ancestry with all the caveats, but that's a lot to look at. And then when we focus down on admixture mapping, we have really a handful of very intriguing results. So we should be able to drill down vertically a lot more on that. We just have not had the opportunity to do so.
And be able to take out the nulls.
Yes. Yeah.
Refining the null signal is interesting. Finding signal that is independent of the null signal is really interesting.
Yes, I had not considered that. That's a good idea. Thank you.
Any other questions from the audience here? Go ahead.
Great talk, thank you. Just quick question. Thanks for introducing RAI. Does it also perform local ancestry inference? Because I imagine for the admixture mapping, you must have.
It does not. And so the algorithm that almost everyone uses for local ancestry is called RFMix. But it was developed in 2013, and it's just too slow and expensive to work with all of us because all of us is in the cloud. There's a couple new ones. One is called Genomics, and we can talk after if you want to do this. Genomics, like Gnome with a G-N-O. And then that is unpublished. That's from Carlos Bustamante's group. And another one is called Flare, which is from the Brownings. That is published. We've been using Genomics in all of us, and it works pretty well. And we also have a bootleg hacked version of RFMix that takes out an expectation maximization step that makes it go fast. So RFMix works great if you have 100 or 1,000 samples, but at the biobank scale it doesn't. So if you are interested in local ancestry inference using all of us or any biobank scale set, we've been flirting with the idea of developing methods, but they're not ready for prime time, but there are others that are out there that I could help you find. Yeah.
Thank you.
Yep.
Okay, so let's go to some questions from the online audience. This question, if the whole world, entire population could be represented, no longer a sample, there would still be peaks or density clusters, yes or no?
Yes, there would, because it would look very, very different. So first of all, almost all of the diversity would be found in Africa, right? So you would have at least two very deeply branching African groups, the Khoisan and the rainforest hunter-gatherer. And then you would have, West African, and this is where really the relationship between genetic diversity and these folk racial categories totally break down, right? So a Nigerian and a Swede would be more genetically closely related than either would be to a Khoisan or a rainforest hunter-gatherer. So it would look totally different. But there would be, there certainly would be continuity across geographic space until you run into a geographical barrier like a mountain range or a desert or an ocean. And then there are also cultural barriers. So humans, we mate assortatively, right? So one of the biggest factors for assortative mating is education. People with the same levels of education tend to mate together. So there's always population structure. Population structure is the rule for human genetics, human population genetics, not the exception. So you would have a very, very different view of global diversity would not correspond at all, because these categories, back to Larry's question where we were saying the same thing, these categories they change over time, they change across countries, but they're based on the pattern and the history of immigration in the country, right? So the categories represent the people that came. At the global level, that would totally break down.
Great. We have another question. This is, how do you decide to draw the line between population groups without reinforcing concepts of the one-drop rule and blood quantum? How does using continental ancestry groupings help resolve this controversy when race and ethnicity share the same geographical boundaries as these groupings?
Right, so first and foremost, I'm under no illusions that our work on genetic ancestry can resolve these long-standing questions. But as it relates to the one-drop rule, the rule of hypodescent, the point with the continuous...
Can you clarify what that is? I don't know what that is.
Yes, this is a cultural artifact of the U.S. history whereby, it's very different from Latin America, whereby individuals who had quote-unquote one drop of African blood were considered to be black.
Oh.
So this is also known as the rule of hypodescent, right? So this is the idea where you're grouping together people for a very specific, in this case, pernicious social reason, right? So but as it relates, so we're not going to solve these issues, right? I'm under no illusion that we can do that with genetic ancestry inference, but what we can emphasize is the extent of variation within groups and the differences in the patterns of ancestry within groups. So the groups are resolved by unsupervised clustering, but even within the groups, you see very, very big differences, right? So at the ends of this, you could see people with very, very different ancestry patterns. So that's, you know, it's not a totally satisfactory answer, I feel, as I'm saying that, but that's the issue. We're not going to resolve these issues, but by focusing on ancestry as a continuous variable, we can provide a much more nuanced view of the diversity.
Okay, another question.
This might be the last question.
Oh, okay, open. Yes. Good. So, there is a recommendation in the field not to use the very ambiguous term ancestry, quote unquote, and instead use genetic similarity. Do you have any reasons to buck these recommendations?
Yes, I think that ancestry, we all have ancestors, and I think that ancestry has a very clear, understandable, and comprehensible definition. I understand and respect that recommendation from the NASM, but I disagree with it. And in general, you know, Steven Pinker has this term, the euphemism treadmill, right? So there's this euphemism treadmill where we're constantly inventing new words to say the same thing, and the things we said before may or may not be offensive at some point. I get it to some extent, but I disagree. I think ancestry is a good word. It explains exactly what it is, and as I said today, it's very relevant for health disparities. And I think that to deal and grapple with these difficult issues, we need to speak plainly and directly. And I think euphemisms take away from that clarity.
Great. So if sickle cell is the positive control, what phenotype would you use as a negative control?
That's a very interesting question.
Yeah.
So, you know, for the admixture regression, I don't know that, oh wait, for the admixture mapping. So something where, so I would say like something like substance abuse, where at first glance you see an ancestry association, but it's, you know, clearly and environmentally determined. There's also an issue of reverse causality there without getting into that too much. But cases where you see a very strong attenuation between the effect size for the ancestry association when ancestry is modeled alone compared to when ancestry is modeled including social determinants of health. So the short answer is substance abuse would be a negative control there. I had not thought about that. I'm going to go back and take a look at that. It's a good question.
There's a lot of online chat with 145 people online.
Yeah. So on behalf of the Genomics and Health Disparities Lecture Series co-sponsors, please join me in thanking Dr. Jordan for a fantastic lecture and for sharing his work and perspectives. This was a timely and needed presentation. I know we have learned a great deal from you today. Thank you for those in attendance and for us, for the people that participated online as well. And thank you for a robust conversation. Enjoy the rest of your afternoon. Thank you.